So a lot has happened over the past 2 weeks. We had Halloween weekend, we had a crazy wintry storm, we had Fall Fest here at Clark, and of course, I did a lot of science. Halloween is one of the most widely anticipated weekends of the entire year, and that's because everyone dresses up and it's pretty great. For instance, this year, I was the Calvin half of Calvin and Hobbes. However, a crazy wintry storm over the weekend kind of ruined and put a damper on Halloween. Not many people were going out, and well, for good reason. On what would have been the primary Halloween night, Saturday, we had a lot of snow coming in. It was this beautiful, windy, wet, wintry mix that brought down a lot of trees. I made some videos here and here showing the destruction on the Clark campus. A lot of the trees were brought down, either entirely or just by losing some arms and legs. And even though the grounds crew has been hard at work, there's still a fair amount to be done. In and around this cold and snow though, we have managed to get out some good weather.
Like today, it's 57.
This past week, we had our annual Fall Fest. Fall Fest presents an opportunity for students to share their projects and research with the administration, professors, and other students. Because Fall Fest is an undergraduate event, I did not present this year, which I am completely fine with because I've had no time to put together a poster and present. In addition to working on this blog, working at the bookstore, I don't have as much time as I would like to work in the lab. So anytime I do have to work in the lab, I am a very, very busy student. In my last lab video, I discussed how I was performing experimental assays on my genetically transformed diatoms. Those assays yielded frozen cell samples, which I then needed to analyze. And that's what I've been working on recently in the lab. Essentially what I want to do is take the RNA out of those frozen cell samples and make DNA from it. And then I can quantify that DNA to give us a sense of the gene activity of the cells at certain points in environmental conditions. And that is the overarching theme of my project, how different genes are responding to environmental conditions over time. So to clarify, certain kinds of RNA are precursors to protein. To make protein, DNA is transcribed into mRNA, which is then translated into protein. And then that protein goes through a special folding and assembly phase to become the active protein and work in the cell. So we want to extract the RNA that's present in the cell to use in our further experiments. While a single RNA extraction wouldn't take that long, I'm usually doing about 8 extractions at a time, which certainly adds up in lab work. A lot of this RNA extraction work is done in the fume hood because one of the chemical reagents I use has a sulfur compound in it and it smells terrible. After a while, the smell really gets to you and makes you feel quite uncomfortable. But in the fume hood, the smell is not really a problem. So in the fume hood, I am washing off the cells from their frozen filters with the smelly buffer. I then take that cell and buffer solution and add it to a small vial with beads in it. These are very, very small beads that we can reuse in RNA extractions. The buffer, cell, and bead mixture goes in a small machine which helps beat up the cells before the actual RNA extraction process. This will increase our RNA yield at the very end. After we beat up the cells, it's very much like a plasmid prep, which I've done hundreds of times. So it's a matter of adding the cell solution to a little filter tube and filtering out the DNA and cell chunks from the RNA, and then adding buffers and solutions to purify the RNA. To make DNA from RNA, we use an enzyme called reverse transcriptase. Reverse transcriptase is an enzyme that allows some viruses to function. They can turn RNA into DNA and make protein and then take over a cell. A reverse transcription reaction is quick and easy and yields us cDNA. cDNA stands for complementary DNA, and we call it as such because we're basically turning the RNA into DNA, so we're just adding a complementary strand to that RNA. Hence the C, complementary. We're still working on ways to quantify the cDNA that we've created with maybe normal PCR or real-time PCR. Real-time PCR will give us a numerical measurement of the number of copies of DNA at each step of a PCR reaction, thus allowing us to better quantify how much DNA we had to start with, or rather RNA, because we turn the RNA into DNA. The real-time PCR reactions should give us great data to support my thesis or help me write my thesis. So that's where I'm at in my project. So over the next few weeks, I'll be doing a couple things. One of the things I'll be doing is preparing more cells to be tested. These replicates will strengthen our data. And actually doing replicates for my project is especially important because of a couple different factors. One, when our plasmid DNA was integrated into diatoms during the transformation process, they were integrated randomly in the diatom's genomic DNA. So this means that our reporter gene GFP might be expressed differently depending on where it was integrated. So this will mean that I'll be doing a lot of ammonium assays, RNA extractions, and generating more cDNA. And as samples come in, I'll be doing real-time PCR reactions.